tenemos el gusto de tener a Paula aquí dándonos esta conferencia acerca de solífugos. Y bueno, Paula recibió su doctorado en 1995 en la Universidad de Florida. En 1998, ella empezó su posición como curadora en el Museo de Denver de Naturaleza y Ciencia. Y su investigación se enfoca en la biodiversidad, taxonomía, sistemáticas y ecología evolutiva de los arácnidos, particularmente en arañas y solífugos. Ella ha publicado numerosos artículos científicos y de divulgación acerca de arañas e insectos. Ella ha sido muy activa en la Sociedad Americana de Aracnología y la Sociedad Internacional de Aracnología, donde ha fungido como presidente de ambas sociedades. Y actualmente Paula es secretaria de la American Arachnological Society y un miembro de la International Society for Arachnology, y pues actualmente es curadora de Zoología de Invertebrados en el Museo de Denver. So, Paula, if you want to begin.
Yes. Gracias, Daniela, y gracias por invitarme a hablar sobre nuestra investigación sobre los solífugos. Yo no puedo hablar bien español, hablaré en inglés, pero incluiré descripciones en español. Y perdón por cualquier error. So today, as Daniela said, I will present some information on the work we have done on camel spiders, a bit about their systematics, and quite a bit about their biology, what we know of their biology. So Solífugos are classified in the kingdom Animalia, in the phylum Arthropoda. They are a class Arachnida or Solífugos. They are important predators in desert habitats throughout the world, eating anything they can get, any arthropod that they can capture, they will try to capture. They are not found, no in Antarctica, no in Australia, but in otros habitats, dry habitats, xeric environments. Now, where Solífugos are placed within the class Arachnida is still uncertain. So there are some studies, some phylogenetic systematic studies that place Solífugos as the sister order to the Palpagradidae, but there are others that place them as the sister order of Raciniolii. There are yet other studies that place them as sister order to the Pseudoscorpiones, and yet other studies place them as most closely related to the Acari, specifically Parasitiformes or Acariformes. So we still do not know where they fall within that large class Arachnida. There are 12 different families in the order Solífugos found throughout the world. In North America, Central America, South America, there are 4 families. There are the Amitrechidae, Dacidae, Aremibatidae, and Mumiciidae. Here in the United States and down through Mexico, we find just Aremibatidae and Amitrechidae. And Amitrechidae becomes even more diverse as you get into South America. And in South America, you find Mumiciidae, and you find some species, 3 species of dacids, but it is uncertain whether those truly are dacids or not, mostly because we do not know if Dacidae is a good family, is a monophyletic family. So we'll talk about that a little bit in a moment. My laboratory is studying primarily the biology of one family, of the Aremibatidae. But in the talk, I will try to give an overview, information on more than just the Aremibatidae, on other families as well. So very, very early on, I'm on my second National Science Foundation grant to study these animals. And in the very first grant proposal, my colleague Lorenzo Prendini used a multi-locus approach, molecular approach, to try to understand a little bit about the entire order Solífugos. But he didn't use very many taxa. He was using just a few loci. But in this very, very preliminary analysis, we were trying to understand if the 12 different families, if each of them were monophyletic. And using this very preliminary analysis, what we found was that it suggested Aremibatidae is monophyletic, and other work that I have done has substantiated that, has indicated that yes, Aremibatidae is a good family. Galeotidae is probably monophyletic. Amitrechidae is a good family, but Mumiciidae are placed within the Amitrechidae based on this very, very early analysis. Solífugos seem to be a good family, a monophyletic family. But the placement and the monophyly of all the other families remains to be determined. And in fact, much work remains to be determined on all of these different families. In particular, look at the Daisidae seem to be polyphyletic. So that may turn out to be an artificial family. But this again is very preliminary, and we need more people. We need someone else to reinvestigate this entire relationship of the 12 families within the entire order. So if we look just at the family that my lab studies, the family Aremibatidae is divided into two subfamilies, subfamily Aremibatinae and subfamily Therobatinae. And those subfamilies had been determined based upon the number of claws on the first leg, the tarsal claw on the first leg. So in the Aremibatinae, these 5 genera, Aremibades, Aremicosta, Aremirax, Aremithera, and Horribades, were thought to have one reduced claw on the tarsal segment of the first leg, whereas these 3 genera, Chambria, Aremichelys, and Hemerotreca, were thought to have 2 claws on the first tarsi. And that was how they were divided up. That we're exploring how true that is in my lab right now. But if we look at these families, we see that 3 of the genera within the family Aremibatidae, Aremibades and Aremichelys and Hemerotreca are very large genera. They make up many, many species. And within each of those 3 genera have been divided into these species groups. So what we did in mid-2000s, this came out in 2015, is we used a multi-locus molecular approach to explore the family Aremibatidae and to understand whether the family was monophyletic, whether the subfamilies, Therobatinae and Aremibatidae, were monophyletic, whether the genera, those 8 genera, were monophyletic, and whether the different species groups in those 3 very large genera are monophyletic. And in this work, what we found is that those 2-clawed genera are probably not monophyletic. They're coming out many places in the molecular phylogeny. Whereas the Aremibatidae, the 1-clawed groups, are mostly coming out together. And some of the genera seem to be monophyletic. So we have Chambria is a monophyletic genus. Aremicosta is monophyletic. Aremirax is monophyletic. Aremithera is monophyletic. But not surprisingly, the very large genera that make up most of the species, Aremibades, Hemerotreca, and Aremichelys, are catch-all. They are not monophyletic. So we have lots more work to do to understand the relationship within those genera. And the species groups are coming out, some of them good, some of them bad, very different relationships. A few of the species groups, Hemerotreca banksii, this group, this group, this group, this subgroup seem to be monophyletic, but most of the others are not. So in conclusions, we found that the Aremibatidae is a good family. It's a monophyletic family that supports that earlier phylogeny that I showed you. Of the 8 genera in the family, many of them, not most of them, but many of them are probably paraphyletic or polyphyletic, except Chambria, Eremothera, Eremocos, and Eremirax, which seem to be good, solid biological entities. They're monophyletic groups. The genera with most of the species, Eremobades, Hemerotricha, and Eremochelys, we need a lot more work done on those. And the species groups within those three big genera are mostly paraphyletic or polyphyletic, probably artificial. So what we're doing in my laboratory now is I have 3 graduate students, Erica Garcia, Ryan Jones, and Goran Shikak. And those 3 students are using next generation sequencing and a lot of morphological characters to better understand the relationships among genera, species within each genus, etc. So much of this morphology that I show here should be familiar to you by now, I would hope. We know that these are arachnids, so they have 2 body parts. They have 4 pairs of legs. They have a pair of pedipalps. They have these powerful chelicerae. In Solífugi, the first pair of walking legs is very tiny and is not used for walking. It is used primarily for sensory perception in the environment, as are the pedipalps. The pedipalps are covered with sensory setae and are held above the animal as it walks in the environment and used to detect sensory signals in the environment. The pedipalps also at their tips have organs called suctoral organs, and I will talk about the suctoral organs and their function in a moment. So there are many different morphological characters that are associated with Solífugi that are very unique. Many characters that are associated with the chelicerae, the jaws, and you should, I think that by now you know that the top part of the jaw is called the fixed finger, and the bottom part of the jaw in English is called the movable finger. So this is very important for Solífugi for many reasons. The Solífugi also have these structures called malleoli on the underside of the abdomen, and we'll talk about the function of the malleoli in a moment. They also have many sensory setae associated with the pedipalps. They have specialized spines associated with the pedipalps, and they have these odd setae, very stiff setae called tenidia on the underside of the abdomen. So my lab is exploring the functional significance and the morphological significance of all of these different structures. But let's talk a little bit about how they develop. I think maybe you may have already heard this, maybe not. But the female, Solífugi female, will lay their eggs in underground burrows. When the eggs hatch, they're in this first instar larval post-embryo stage. And these larvae will clump together, and they are pretty immobile. They just aggregate and stay in a little cluster, but they don't move very much. They are persisting on any nutrients left over from the eggs. Then they will go into the first instar nymph, and at this stage, we begin to see some pigmentation, a little bit of sclerotization, pigmentation. By the second instar, they are much more mobile, and they are hardened, so the pigmentation is much more evident. The jaws are hard, and they can begin to hunt and to prey upon little insects or upon each other. So they become little cannibals at that stage, which means that if you try to raise them, you must separate them if you don't want to just end up with one very large juvenile. And then by the third or fourth instar, they become yet more active and are out there hunting and digging in the environment. In these juvenile stages, you know that they are juvenile because the genital organs are not formed, and because of those structures that I call malleoli. The malleoli on the underside of the abdomen adults have 10 malleoli, 5 and 5, whereas the juveniles only have 3 malleoli on each side, on the underside of the 4th leg. So what are those malleoli? This is what they look like under the microscope. They are these leaf-like structures on the underside of the legs, and they function as chemoreceptors. As the animal moves through the environment, it drags the malleoli on the ground, and they pick up chemical cues from each other or cues from prey. Other characteristics that are important have to do with the chelicerae. So in the family of Aremabatidae, the male chelicerae look quite different than the female chelicerae. The male chelicerae in most of the genera have no teeth on the fixed finger, whereas the females have teeth on both the fixed and the movable finger. And we see other pretty dramatic modifications in the male chelicerae. That's in the family of Aremabatidae. In the other families, we see other modifications. So in other families, the non-Aremabatidae, we see these structures called the flagella, associated with the fixed finger of the male. We do not know the function of the flagella, but we suspect it is involved in copulation, in mating, in courtship, because it is only found on the males. And this is a plate from Tarina Bird's very nice monograph showing all the different structures of flagella in the family Solifugos. And in this family, the flagella are species specific. And again, we do not know the function, but they're important diagnostic for species identification. The animals also have these organs at the tips of the pedipalps called suctoral organs. We see an SEM of that suctoral organ right here. And this is another view of that SEM. This suctoral organ is everted, is been popped out of the tip of the pedipalp. So the suctoral organ functions, it functions much like the toe pads of geckos. The structure itself acts to hold on to smooth surfaces, like the smooth body of an insect or the body of the females. The males will use the suctoral organ when they're mating, and they use those suctoral organs. They evert them out of the pedipalp, and they hold on to those smooth structures. They can use the suctoral organs to pull prey close to the chelicerae. So in early 2000, my lab did some studies on looking at the functional significance of the suctoral organ. And what we found was that the suctoral organ is expanded, is retracted into the pedipalp using ligaments that pull the suctoral organ into the pedipalp when it is not used. And hemolymph pressure, increased hemolymph pressure will expand the suctoral organ out of the tip of the pedipalp. And the male Solifugos, whatever family we're talking about, will use the jaws, the chelicerae, and the suctoral organs when mating. Or we're not sure about the suctoral organs in all the families. We know they use the suctoral organs mating in Aremabatidae, and it is likely they also use them in other families to hold on to the female. So in mating, the male will find the female, probably using pheromones of some sort. He locates the female, he grabs hold of the female, he contacts her, and as soon as the male makes contact with the female, she immediately stops moving. We have no idea what signal causes that cessation of movement, but it is apparent in Aremabatidae, and it seems to happen in other families as well, where the contact by the male causes the female to stop moving. The male then uses his suctoral organs to maneuver and manipulate the female's body so that he can insert the fixed finger of his chelicerae into the female's gonopore. We do not know what he is doing, but he's chewing. It's chewing motion. And we think maybe he is preparing the female to receive his sperm, or he is removing competing male sperm, or both. In Aremibatidae, the males will then remove their fixed finger from the female, move their abdomen up, directly deposit the sperm packet onto the female's abdomen, onto her gonopore, move his abdomen down, and then he uses the fixed finger to push that sperm packet into her body. And then he chews again, and that chewing releases the sperm into her body, into the sperm receptacles. We see the same kind of mating in other families as well. But in some of the other families, like Galeotidae and other families, the male does not directly deposit the sperm packet. Instead, he pulls the sperm packet from his abdomen, from his opening, and then places it into the female. Once he's placed the sperm packet, the female very quickly begins to wake up. And when she begins to wake up, he books it out of there. He runs and releases her and gets away before she can eat him. So a student who I helped named Jen Rousell did this nice video showing mating in Aremibatidae, and you can see all of those same stages that I just described. So this that we see here is the female, and she's running around. This is a mirror on the bottom of the terrarium, so you can see underneath the animals. This is the male coming in. He contacts her. Now watch the female. She will almost immediately begin to stop moving. So she has become completely immobile, quiescent. He's using those suctoral organs to move her body and to open up her gonopore, bend her body over. He sticks his fixed finger into her. He begins to chew, and he chews, and he chews, and he chews and chews and chews and chews. This goes on for over a minute. So this is going to be for a while. And again, we do not know what's going on here. We suspect he is preparing her to receive his sperm and or removing competitor sperm from her genitalia. But very soon, at some point, sort of soon, you will see him remove his chelicerae, move his body up, deposit the sperm packet, and then move his body down and push the sperm into her. But right now he's still chewing and still using the suctoral organs to move her body back and forth. Now he's about to move his body up. Releases, deposits the sperm, then pushes it back into her. Now watch the female, because she will begin to wake up. She'll begin to move her jaws, and she'll begin to move her legs. She's beginning to move her third leg here. And now you can see her jaws moving. And once she becomes even more active, he will very quickly release her, and they will separate. I probably will not play this all the way through to the end, however, because she's becoming a lot more active. Let's just skip ahead a little bit. Ahead a little bit more. He still wants to hold on to her, but she's trying to bite him at this point. And now she's trying to get away. She's got her jaws moving, and she's desperately trying to get out of his grip. And finally, he separates. So the last part of the lecture, I just wanted to give an overview of some of the other morphological characters that we have examined in my lab. And this is all towards understanding the function of these structures as well as their importance in taxonomy. So we did a small study looking at some of the many different kinds of seta that cover the pedipalps of Solífugos. And we see setae that have openings that probably are chemoreceptors. There are others that are bifurcated that we see in almost all the families. And these are probably, who knows, mechanoreceptors, we're not sure. There are also these very strange little structures that look like trees that are called papillae. And papillae are important in diagnosing or identifying genera and species. So we did an investigation of these setae. And the papillae, this is what they look like. They're on the underside of the pedipalp. And in SEM, they look like little tiny trees. They are found in 3 of the 12 families. They are found in the Aremabatidae, only on males, Cartiidae, only on males, and Solífugos, only on males. So they may be picking, we hypothesize that they were chemoreceptors or mechanoreceptors somehow involved in mating or copulation. You can see that the structure looks quite different depending on the family. So all the Aremabatidae species that have papillae, they all look like this. All the Cartiidae species that have papillae, those papillae look like this. And all Solífugos species, papillae look like that. So we hypothesize that they may be mechanoreceptors, chemoreceptors, olfactory receptors picking up airborne chemical cues, secretory organs that are producing some chemical signal. So we used transmission electron microscopy and scanning electron microscopy and electrophysiological experiments to test these different hypotheses. And using these techniques, this is a cross-section through a papillae. In our TEM results, we found that there is a dendrite that extends up through the shaft, the center of the papillae. So we can see that dendrite here. And that dendrite is characteristic of arthropod chemoreceptors. We also saw pores, openings to the outside on the branches of the papillae. And our TEM work also demonstrated this is a cross-section through the base of a papilla. And we saw dendrites called tubular bodies that are characteristic of mechanoreceptors. And we also did electrophysiological experiments where we blew different kinds of signals, of chemical signals across the papillae to see if we got a chemical, or an electrical signal, a response from the dendrite. And this is what we believe that the structure looks like, where you have tubular bodies at the base, we have a dendrite going up through the center, and there are pores, openings to the outside of the papillae. The data, the electrophysiological data and the TEM and the SEM data suggest that these papillae function as both mechanoreceptors and chemoreceptors. They emerge from a pore at the base on the surface of the pedipalp, and the mechanoreceptors, the tubular bodies are at the base there. The dendrites extend through the shaft of the papilla. The pores, there are pores that open to the outside that pick up chemical cues. We did not find support for the hypothesis that they were secretory organs. And we did not find support that they were thermoreceptors picking up temperature cues or hygroreceptors picking up humidity. So it seems like they are chemo and mechanoreceptors. And that was a publication in 2014 that came out. Erica Garcia has been examining these diagnostic spines that are on the pedipalps of Solífugi to explore their function and their importance in identification, identifying different species. And Ryan Jones has published a paper looking at how the diagnostic importance of these ctenidia that we find on Aremabatidae, but we find these structures in other families as well. We do not know the function of the ctenidia, but they're found only on males. So I don't know the function. We don't really have a good working hypothesis. And this paper came out in 2021. What Ryan found is the number of ctenidia, but not the shape or the length, have some signal, some important signal phylogenetically. But again, functionally, we do not know. Erica, in her talk during this symposium, already spoke about how we collect Solífugi. But we do use lights. So we know that Solífugi are attracted to lights, where insects are also attracted to lights. The Solífugi come to the lights to hunt the insects. So this is our typical trap design, where we have pitfalls set up underneath suspended lights that will attract the Solífugids to the pitfall traps. And in our project, I want to point you towards our website. If you go to www.solífugi.info, we are aggregating as much information as we can about the biology, the taxonomy, biogeography, research on this order. So I encourage you, if your lab is working on this group, that you should send information to Warren Savory. His email is here. He would very much appreciate any content that he can incorporate into this website. So please consider that. Our data, our collections data, is being input into this online database, the Scan Symbiota database. And this is the URL for that database. We are also building an online field guide to the different species of Solífugi found in North America. So I point you to this site as well. It's very useful. We are trying to incorporate into this field guide photographs that are useful for identification. So photographs of the chelicerae, photographs of the color patterns and the dorsal and ventral morphology, photographs of the genitalia, female operculum, all sorts of photographs that you can use for species identification are being posted there. And through this same site, you can also create species distribution maps using the scan data that will give you an idea of the distribution of different species throughout North America. This is an example of one of the data pages, so you can also get a lot of information, detailed information about the exact locality where species were collected. And I just want to say that I am very excited that in recent years, there have been a lot of really excellent publications and research being done on this group of arachnids throughout the world. So it is an exciting time to study these arachnids. And yet there's a lot we still need to understand about this group, about their natural history, their biology, their distribution, their systematics, their behavior. So if you are very interested in arachnology and are trying to find a good group to study, I would encourage you to study Solífugi. And with that, I want to thank you.